unexpected. So, but finally we are here, and our colleagues online are also waiting. So, may I introduce Dr. Imran? Is working as principal scientist at the National Institute of Biotechnology and Genetic Engineering. He's I've been working with me right from the beginning, when he was an MSc student. So he's a very close and long association. Dr. Ibn Amin basically got his PhD degree in characterization of viruses and suppressor of gene silencing encoded by some of these plant viruses, specifically plant germ viruses. And so one of the approach that we have is to have gene silencing, and then when we so we use different technologies. I myself got very welcome on mRNA technologies, but now we have developed these new technologies for silencing or mutating gene by using genome-editing technologies. So we will talk about this topic, which is more related, about designing of these tools. Besides designing tools, we also work on different versions of Cas proteins and their applications, and you might have seen publications coming up from our lab, basically to develop new tools. Thank you.
How to share? Can you? Good morning, ladies and gentlemen. Thank you very much, Dr. Shahid, for the introduction.
Yes, just share the screen. And this.
So I've been asked to sit here because we have virtual participants as well. It should work now? No. Understand this.
Okay.
Presentation mode. Do we have a presenter?
We don't have a presenter. Can you use the one?
Don't have this. Stop share. Share whole computer. And
okay, it's sharing.
Share this one.
Dr. Imran, I think अब मेरे फिलहाल मोबाइल से ऑफ कर रहे हैं. Can you ramble on for some? Can you ramble on for some time? Because इसमें मेरे जो virtual वाले हैं उनको दिक्कत. चलो.
Thanks.
Okay, the screen is shared, right?
Now we can share the screen.
Sorry, what is this?
Yeah.
This is not internet.
You need to. You need to share the screen. Share
Yeah,
the
yeah.
screen on.
Yeah, don't worry, I will do that.
Share presentation. Okay, sorry for the delay. There was a technical problem.
Is it possible to move?
Oh, yeah.
Not really.
Yes,
Oh,
it
yeah,
is.
yeah.
It is. So, first, let me apologize for this delay. And then secondly, what I think is that it should be a practical session rather than a lecture, but unfortunately we were unable to manage that. So I'll try to manage it with some screenshots. I will share some web pages. I'm here for 3 days, and if anyone is interested, we can work on the computer and we can work on their guide RN. So the content, this is not a research topic, it's basic. So I'll discuss what is genome editing, of genome editing, because that is required for designing your guide RNA. Then a brief history of CRISPR-Cas system, and then toward the end, we will discuss in silico guide RNA designing for CRISPR-Cas system. So if you talk about CRISPR, it's a bacterial immunity system first discovered way back in late 1788. It is working against bacteriophages. It is a very conserved mechanism. It is found in 40 and about 90% of archaea. Is that is a tree interspersed palindromic repeats. It is the CRISPR is direct nucleotide repair, which are separated by the and then there is a protein associated protein which use those guide RNA as a it results in the cleavage of the DNA. Naturally, what happened is that when bacteriophages infect a cell, that it will inject that cell. That DNA like this. If this is the DNA, it is recruited into the CRISPR array. So what it does is that whenever there is a secondary infection of that, this DNA is acting as a guide. It will, and that RNA will guide this Cas protein, and it will cleave that bacteriophages. So there is no secondary infection, theoretically speaking. How we use this system in I have given the example of the plant is like that we develop our own guide RNA. We develop, I'm talking about, we develop the construct of Cas9 as well. And then we transform that into the host cell. In the host cell, the guide RNA is and then this Cas9 is produced. This guide RNA will guide this Cas9 protein, and it will, of course, there is a sequence which is called as PAM. I'll discuss this in detail in the later slide, that it will result in the cleavage of the DNA. Okay, another thing is that when we talk about the repair of the DNA, the cell has this natural ability that it can repair itself. There are two kinds of non-homologous result in the indels. We call it insertion and deletion, and then there is homology-directed DNA is the knockout. So this genome editing is a technique where mutation, you can also produce the knockout gene. So it can be used in the synthetic biology. It is used in gene therapy, and there are certain Cas proteins which are used for the gene regulation as well. If you see that in my career, observed is that this is one of the technology that got prize in a very little time. The first one, if I remember, that was the Caramulus, the PCR, and then the second in biological sciences is this. That this era of divided into three. The first is identification phase, and then the characterization, and then its application. The applications are huge, not only in plant sciences, it is in the animal sciences as well. Now I'll discuss very basic, which is required for designing about CRISPR-Cas9. Let me first tell you that there are around 90 Cas proteins, and each Cas front, it has its own cutting mechanism, how it cleaves. Its requirement is different, so you have to take care that which Cas protein you are talking about, design accordingly. The most widely used protein is Cas protein, is Cas9 protein, which most of the time we work with the Cas protein. The class 2 type 2 system. This is very important. This is protospacer adjacent motifs. We call it PAM sequence. The Cas protein require a PAM sequence to cut down or to be guided by the guide RNA. In case of Cas9, this is NGG, but for every Cas protein, PAM sequence is different. Then there is a sequence we call the seed sequence. If you see here, this is basically the guide RNA. The guide RNA size of the Cas protein is 20 nucleotide. The seed sequence is, this is PAM sequence, and this is the guide RNA. That's proximal sequence, which from this PAM sequence up to 11 nucleotide is basically the seed sequence. Seed sequence is very important. If you remember the previous lecture where Dr. Hensel was telling you about the off-target guide RNA. This sequence is important because if there are mismatches in this sequence, Cas protein can tolerate that. If there are mismatches proximal to the seed sequence, Cas protein will definitely cleave that, and it work on, it will produce its effect, what off-target effect. Then there is a CRISPR RNA. If you see, this is the guide RNA. Upstream of the guide RNA, there is another RNA which is called as CRISPR RNA. This CRISPR RNA basically binds with the tracer RNA. Our functional guide RNA constitute the guide RNA as well as the CRISPR RNA. CRISPR RNA bind with this tracer and it form an arm that on which Cas9 detect not only the PAM, but it also detect this tracer RNA and it act on that, and then it result in the cleavage or whatever function we wanted to have. I'll first tell you different Cas system on which we are working, and then I'll discuss about the designing of RNA. This is CRISPR-Cas12a, we call it CPF1. It is class 2 type 5 system. The size of the CRISPR protein is around 1300 nucleotide. And by the way, Cas9 is also 1300 nucleotide. It is capable of processing its own CRISPR RNA, and it uses only ROV-C domain. It's basically the domain that is responsible for the cleavage of the DNA. Cas12a does not require any tracer RNA, so this is different from the Cas9. That there is no need of tracer RNA for that. And the size of the guide RNA is around 41 to 44 nucleotide. In case of Cas9, it was 20 nucleotide. In case of this Cas, it is 44 nucleotide. The PAM sequence is basically 5' TTN, which is, N is for any nucleotide, which is located directly upstream. The main difference between Cas12 and Cas9, sorry, if you see this, the guide RNA is on downstream. It is on the 3' end. In this case, it is on the 5' end. So every Cas protein has its own guide RNA, the size, and its direction, whether it is present on the 5' end or it's present on the 3' end. Another property of this CRISPR-Cas12 protein is that the indiscriminate cleavage of collateral single-stranded DNA. Once it is activated by the guide RNA, it worked on the double-stranded RNA. But when it is activated, it indiscriminately cleaves the single-stranded DNA. And this is very important in a way because there are certain viruses which are single-stranded, and we cannot target single-stranded viruses, though we have another Cas protein that can target that, but it can target double-stranded RNA. It can also target the single-stranded RNA. This is another system of the CRISPR, which is CRISPR-Cas13a. This is previously it was known as C2C2, and it is class 2 type 6 system. It is an RNA-guided RNA editing system. The previous two Cas proteins which I was discussing, those basically cleave the DNA. This CRISPR-Cas13a, it cleaves the RNA. So we have both the proteins that can cleave the DNA as well as the RNA, and Cas13a is the one that can cleave single-stranded RNA. It has dual function of CRISPR-Cas13a system. Pre-CRISPR RNAs are processed into the mature, so its CRISPR RNA is produced by itself. The size of its guide RNA is around 28 nucleotide. This is another system, which is CRISPR-Cas14a. This is relatively a smaller protein of about 400 to 700 amino acids. The size is 40 to 70 kDa. Another property of this CRISPR or Cas protein is that it don't require PAM sequence. So if you give it a guide RNA, it will result in the cleavage. And the cleavage is basically of the single-stranded DNA. It don't restrict or it don't cut the double-stranded DNA. And the guide RNA size is 37 nucleotide. Now let's talk about up here. The point which I want to make is that we have different kind of Cas protein. You need to know which Cas protein you are working with, what is the requirement, what is the PAM sequence, and then whether that PAM is present upstream or it is present on the downstream, and then you design your guide RNA. If you talk about the summary, it's a whole of the genome editing. That first thing is you need to know the sequence. Mostly either you download that sequence from any sequence database services, but we prefer to have sequence of the crop or the plant which we are working on, so that you have exact same sequence and you can design the guide RNA. Then there are certain software which I'll let you know, but I'll tell you there are several the software which we use during this. And we also, by the way, I'll also tell you how we manually design the guide RNA. When those guide RNAs are designed, and one of the most important part is to know that whether they are going to produce off target or not. You know, the size of these guide RNA is around 20 nucleotide or 40 nucleotide, and this homology, you can find it anywhere. In humans, in plant, this is less problem, but when you talk about the mammals or the higher animal, it is a problem. So you need to you design your guide RNA in such a way that they should not produce the off-target effect. Once you get that off-target, you find that good guide RNA, you clone it. We use Golden Gate cloning in that, and then you transform it, and then you do the sequencing. I think the previous lectures has shown you that how we do that. So guide RNA for the Cas9, for the example, I'll show you the Cas9. First of all, if you see, this is the PAM sequence, and the guide RNA is the 20 nucleotide which is present downstream toward the 5' end of this. So this will be the guide RNA, and this is the PAM sequence. The PAM sequence itself is absolutely required for cleavage, but it is not part of the guide RNA. The PAM is not included in the guide RNA. We design guide RNA without the PAM sequence. If you're talking about mismatches, so the limit is 3 nucleotide. If you have 3 mismatches in your seed sequence, as I mentioned earlier, the guide RNA will tolerate that. The Cas9 will tolerate that, and it won't go for cleavage. But if that homology is, the mismatch is away from the seed sequence, then it will definitely produce the off-target, and you have to take care of that. I'll show you with few examples later. These are the different guide RNA designing tool. As far as their work is concerned, it's simple search. What you need is the PAM sequence, even in the work. So you can design the guide RNA. These are, I'll go through the, I'll show you screenshots. For example, this is GeneScript, and if you see that these are the genomes which are available on this website. These genomes are basically the reference to tell you the off-target, because when you give your input in this in the server or in this web page, they will end up with the guide RNA, and they have at the back end they have these genomes, and these genomes, they will tell you that whether your guide RNA will produce off-target effect or not. This is basically Eukaryotic pathogen CRISPR guide RNA design tool, and there is a lot of flexibility in this. I usually prefer this for designing the guide RNA. What we do is that if you see here, first you have to tell this this server or this interface that what is the PAM sequence, what sort of Cas protein you are working with. Here, these are the different Cas proteins. Each Cas protein has its own PAM sequence. So you first have to decide that which PAM you require, which Cas protein you are working with, and then they have. If you see here, they have different genomes. You can, out of these genomes, you can identify or you can select the sequence, or if you go there, you can also give your custom genome. By custom genome means the sequence which you will give, which you are interested in, and you can have the guide RNA. It will give you an output like this. For example, this is a cotton leaf Codon virus. I just wanted to know that where are the guide RNA. If you see that, it will give you this. This is the PAM sequence. Of course, this is not part of your input, but it gives you that this is the PAM sequence. It will give you the score. The score it gives basically that they have come up with the numbering, 1 being maximum and 0.01 is the lowest, where it numbers, it gives you a score based on the specificity of that guide RNA and its chances to result in off-target effect. It will also tell you that during transcription, what could be the problem and what nucleotide you may have to add so that you can have a proper RNA in that. This is another web page where a web tool which you can use, but it is only for these organisms. But if you see here's the Arabidopsis thaliana, you can use it for Arabidopsis thaliana as well. This is specifically for the stem cell, is for human and mouse genome, where it can work with. Okay, this tool is for CRISPR cereals. There they have wheat, they have rice, both Japonicum and the other one, and then Zeaxanthin. So if you want to work on that, you give your input in this and it will provide you your guide RNA. This is another tool because it has flexibility again. Here you can, they have their genome as well, and then you can also give the custom genome which you want to work with. This is called CRISPR Direct. If you give your input and you have to select what sort of PAM sequence you are looking for, and then based on that PAM sequence, it will give you the guide RNA. If you see here, this was a custom genome, means the genome which I want to work with, it cannot give you the mismatches because that genome was not part of this web page. This is another example where I have given that I want to see that how many mismatches are there in case of human beings, and the same output comes with this, and it can tell you that how many mismatches are there and what are their position, whether they are present in the seed sequence or they are present away from the sequence, from the seed sequence it can tolerate or not. So the thing is that you have to identify the best guide RNA that will suit you. This is another tool which is called CRISPR P2. This is again very widely used. You give your input in that and it will provide you the guide RNA along with the PAM sequence, and then it will tell you the GC ratio and the other stuff. Now, up till now, these are the different other tools which you can use. Designing is not very difficult. It is basically the selection of the guide RNA, which is the best guide RNA that can be utilized. And that is done by if you are sure that it don't have off-target effect. Before that, let me tell you that how we do manually. Manually is very simple, as I mentioned earlier, that you need to know what is the PAM sequence, where your guide RNA lies upstream of the DNA or downstream of the DNA, how many nucleotides are there, and then you can take out that sequence and then see that whether this is going to work, it has some off-target effect or not. This is one of the example, one of our student, she was working on potato. It is basically to develop resistance against potato. She was working on ELF. It is an elongation factor in the, I'm sorry, potato. It was ELF4 is an elongation factor with which PVY, potato virus Y, interact. So if you stop that interaction by mutating the ELF4 gene, there is a chance that you get resistance against those viruses because that virus cannot replicate. So what we did was that we not only downloaded, we also sequenced that. We find the ID of that sequence. And then another thing is that usually you design your guide RNA not on the intron region. It has to be an exon. So you identify the exon region where those exon lies. After those, there are several software. You can do it in any software which you are working with. We highlight those exons, and from those exons, you identify or you find the PAM sequence. And if you're working with the Cas9, you go 20 nucleotide downstream of that sequence as a guide RNA for you. As far as cloning is concerned, really, these are oligos which we synthesize, and we use type 2 restriction endonuclease Golden Gate cloning, in which it produces 4 nucleotide overhang. We add those overhangs with our guide RNAs so that it can be cloned in those particular vector. And I am not going to detail because the next lecture is about those the vector system, and Dr. Zahid probably will tell you about how we do cloning in that. Now, this is the most important part. You have designed, you have the guide RNA, you can have hundreds and hundreds of guide RNA because these PAM sequences, they are NGT sequence. But the problem is guide RNA, and that guide RNA should be the one that you. And in this case, I mean, blast, everyone probably must have done the blast. You can do blast in batches, and you can do 100 of those nucleotides. You can blast each homology, or you can restrict that blast to a certain particular organism. You want to see the off, you can do that blast as. We use several tools. One of the is this this cat. I can web pages with you. So what you have to do is that your input here, the guide RNA, the 1000 guide in one go. So what you do is that Excel file here, and you how many mismatches, up to 3. You have to put, because of that, right here. So once that the organism which you are working with, for example, what I did was Marsutum, and that. Anyways, if you see as an example, this that is giving you effect of target of 1 number, and this is a guide RNA that there are 253 off targets. So obviously you will select the one which has minimum number of off target. For example, this is the detail of that experiment. If you see, this is the seed sequence. I told you that it is about 12 nucleotides. If there is a mismatch here, this can be tolerated. Ideally speaking, there should be no mismatch. And there are certain guide RNAs. It shows only those guide RNAs that shows the mismatches. Those guide RNAs you have to work on a particular sequence, so you don't have this liberty. In that case, you have to see that where that mismatch is, whether it's in the seed sequence. The thing is that it should be in the seed sequence. You can go with that, but if it is not in the seed sequence, then you cannot use that. You have to come up with some other strategies. This is the cloning. That is all. This is basically what I want to show here is that these are the nucleotide overhang. This is the guide RNA to here, 20 nucleotide, and the overhang are introduced just to clone them in the Golden Gate vector. This is in silico cloning, which we usually do once we design guide RNA. There are certain software which can do that, and that is all from for the designing. I would like to acknowledge COMSTECH, COMSEC, and me too, and thank you very much.
Now, I think we should take all the questions at the end of the 3 sessions. That would be better. Okay. So we will continue with the next speaker. This next lecture will talk. We will have the lecturer from Saudi Arabia, Dr. Zahir Ali. Dr. Ali works on genome editing in different crop species, and today he is virtually joining us on this Zoom, and he will talk about the vectors used in the genome editing, and basically he will tell us how to design those vectors. He should be here.
I think he is there. Yeah.
Okay.
Okay, Shamshad, please unmute Dr. Zahir Ali so that he can speak and share his video and slides.
Hello.
Okay, yeah, go ahead, Dr. Zahir. Please turn on your camera as well. We cannot see you. Yeah, now.
Can you see?
Yes,
Yep.
yes. Please also
Yep.
share your slides.
Can you see my slides?
Yes, we can see.
Yep. Yep. Thank you for the nice introduction. I'm Zahir Ali. I'm working as a research scientist at the King Abdullah University of Science and Technology, Saudi Arabia. Basically working with the plant genome engineering and specifically with the CRISPR-Cas system, which is now converting mostly into every field, including genome editing plus diagnostics. Today, I will present some work about the vectors used in genome editing in crops. So I will talk about different vectors that are used in the plant system. Before that, I will talk about as Dr. Imran already presented this one, but I will just briefly introduce that the CRISPR-Cas system is basically the immune system of the viruses, the immune system of the virus, and basically it's a memory system, like when the virus attacks the bacterial genome, the bacterial cell, and the genome of the virus enter into the bacterial cell. The bacteria keep a small piece of this virus, the virus part with itself in the genome as a memory. And this is called like immunization. So now the bacterial, if the bacteria have a small sequence from the virus genome as a spacer, then it can give memory for the next attack. So like, for example, when there is the virus DNA again, or the virus attack, the virus come back to this bacteria, this gRNA or this spacer is expressed in the form of sgRNA, and this make a complex with the Cas9, and then it can target the virus genome. And then after this targeting, the virus genome is destroyed. So this is the immune system of bacteria and archaeas. And very specifically, they are present for the RNA viruses or the RNA phages or the DNA phages. And the system is very simple, like it's need a simple Cas9 or the Cas nucleases, and it need a guide sequence that can go to target DNA. So looking at the system, looking at the simplicity of the system, when these systems were discovered, it's very easy that it can be used for genome editing of any DNA. So like, for example, it's just need a very simple sequence, like you need only 20 nucleotide sequence, followed by an NGG or a POM sequence, which is different for every Cas9 nuclease. And then you can put this into a gRNA sequence, and here is a scaffold which is binding, which is responsible for the binding with the Cas9 protein. So when these two combine, it can come to the target DNA, and then it can make a double strand cut, because here we have the ROF2 domain, and here we have the HNH domain, which make two cuts on the DNA, and the double strand cut is there. So these are the basic requirements for the CRISPR-Cas system for the genome engineering or genome targeting. But then normally how we are using this system and how we can clone this system, normally we are using, there are different methods like the restriction ligation system, the gateway system, and nowadays, because the CRISPR-Cas system, it's very it's need very a lot of cloning and a lot of gRNAs we have to check. So it's need a simple method to clone the things. And one of the simplest methods recently people are using is the Gibson Assembly, which is very straightforward. You just need a vector that even you can cut with the restriction enzyme, or you can do the PCR for it. And then whatever you are cloning, like for example, if we are cloning the Cas9, we need a tag, we need a nuclear localization signal, and then you can just amplify, PCR amplify with a specific sequence overlap, and then you just mix these three things, and then in a single enzyme and at a single temperature at 50 degrees C, 15 to 60 minutes, you can clone all the fragments. Like for example, you can go up to 12 fragments, and even the whole genome of bacteria can be cloned with this method. Just the PCR fragments are there, and then you can make them into a single clone. How it's work? There are three enzymes basically working in this system. One is the 5' exonuclease, which start cutting from the 5' end. And then we have a DNA polymerase, which fill the gaps. Like for example, these two overhangs are there. So the 5' end, it start to degrade this one, and here it start to degrade this one. So then you have a complementary sequence for one another. The same is happening here. And then a gap is there because the exonuclease remove some sequence. So when the gap is there, this gap is filled by the DNA polymerase. And then the ligase. So when the gap is filled, then we need a ligation. So here the ligase will come and it will make the clone. And here the same will happen. And these three enzymes work together at this simple one temperature and one tube. And then you just transform this into bacteria, and you can see the colonies transformation very easily. Compared to the other system, this system is very simple and it's very easy and very robust. Like for example, you can see almost 99% of the colonies, they are positive, and it's very simple compared to the other methods. But this is just about the cloning. All the clonings can be done with this simple method, even though the plant vectors, sometimes they are a little bit big. But even if the vector is big, you can just do a pieces PCR. Like for example, this, if we are amplifying this fragment, so we can do a PCR here, then a PCR here, and then a PCR here, and then we have 5 fragments. So all the 5 fragments can be put in single tube, mixed with the Gibson assembly, and then transform. So you will see a whole clone and then the transformation. So the system is very simple. Then what are the basic requirements for the CRISPR-Cas system for plants? The first thing, the CRISPR-Cas is a bacterial system, which is a prokaryotic system. And it cannot be expressed as it is because it's a very big proteins normally. Nowadays, there are some small proteins discovered in the CRISPR-Cas system, but normally they are big protein. And this much big protein from a prokaryotic system are not able to be expressed in the plant system or in the eukaryote. So the first thing is like codon optimization, because the expression is basically dependent on when the RNA is there, then we need the codon and anticodon system to make the amino acid or the polypeptide chain or the Cas nucleases. So the first thing is like we need a codon optimized Cas nuclease. Whatever, like for example, if we are using Cas9 or if we are using Cas12 or Cas13, whatever, but for the eukaryote, we need the codon optimization. This is the first requirement. The other thing, like there are different Cas nucleases. So which one we want to use? And they have different advantages and disadvantages. Like for example, Cas9 is very robust, but it has some off-target effects. Cas12, it's also working very well. There are a lot different available. So whatever we are using, we have to select very precisely for our purpose. The second thing is like all the Cas nucleases, they don't have any NLS. And the genome, compared to the prokaryotes, the genome of the eukaryotes are in the nucleus. So we need to put the NLS tag with it. And if we want to detect by the Western blot or by different methods, so we need the other tagging like flag tag or the HA tag. We also need these things to be in these sequences. And then the orientation, as Dr. Imran explained in the previous lecture, that the orientation of the guide sequence is very important. And the palm sequence is basically determining the orientation, like where is the palm, and after that the guide sequence will be there. So it's very important to select a very precise orientation of the guide sequence. Then selection of the nuclear expression promoters. This is the most important one because all the genome is there in the nucleus, and specifically the gRNA, like the Cas9, we can put the NLS. But for gRNA, it must be in the nucleus. So if it is expressed from a promoter, which will take it out of the nucleus, like for example, the pole 2 promoter, all the RNA is expressed from it, it's go out. So we need a pole 3 promoter to express the gRNA to keep it inside the nucleus. And when the NLS with the Cas9 is there, and then it can work on the genome for genome targeting. The next thing, and these are the basis how we design the vectors. So for example, for the Cas nucleases expression, we need an RNA polymerase, which is basically RNA polymerase 2. And this is the promoter sequence. And we need a specific sequence, which is called the 5' UTR. And the transcription start from here, but the protein start from here. So this sequence just gave the stability to the protein. Specifically for big protein, these sequences are important. And then we have the coding sequence. Then there is a stop codon, and after stop codon, the protein, the amino acid sequence is stopped. But here we have 3' UTR region, and this is really important for the stability of this much big protein, specifically like Cas9 or Cas12. They are very big RNAs, and these sequences are basically provide the protection. And then the transcription stop site, which is normally we call it terminators. In our experience, the promoter is important, but for the Cas9 specifically, the terminator is also much more important than the other proteins. The next one is the gRNA. So here, as we see that in the gRNA case, the protein case, the Cas9 protein case, we need a specific sequence which is not part of the Cas9, but just for the protection of this express sequence. Here we don't need any extra sequence. Specifically at the 5' end of the gRNA, it's not tolerating more than 1 or 2 nucleotides. So if there is a long sequence, it will not work with the Cas9 and it cannot be used. The same is at the end. Even though the end is this 3' end is little bit flexible, but we should be careful, especially with the N nucleotides that should be multiple T's. Which gives stability to the gRNA structure. And when the gRNA structure is stable, it will be available for the Cas9. Then we have two different, in plants, we have two different systems, like we have dicots and we have monocots. So the basic thing is like when we are doing for dicots, we have to select the sgRNA expression, normally the RNA polymerase 3 promoters, which is normally a U6 promoter, and the Cas9 expression, which is the RNA polymerase 2 promoters. Normally, we are using Cas35S promoter, CAMV35S promoter, or ubiquitin promoter. In our experience, normally 35S promoter is not working that much better. It's better always to use a ubiquitin, which is not very constitutive, very highly expressed promoter, but the promoter or the protein produced from ubiquitin is very stable. Then for the monocot, we need the sgRNA expression. Again, we are using the RNA polymerase 3 promoters. Normally U3 promoter or U6 promoter. Compared to U6, here we are using only U6 promoter, but here compared to U6 promoter of the monocots, U3 promoters are working better than the U6 promoters. The Cas9 expression, for Cas9 expression, we can use these both. But the case, normally people don't want to use the 35S promoter in monocot, so that's why the ubiquitin and specifically the maize ubiquitin promoter is working very well. In the initial system that the CRISPR was developed, the first one was used as a 35S, like in protoplast, and specifically they use the protoplast from two different organisms, in Arabidopsis as well as in Nicotiana bentamiana. And they use 35S promoter to express the Cas9, and they put like the NLS and the flag tag just to detect these sequences, this protein produced. Also, they put an intron just to provide a stability into the Cas9 mRNA. And they put the NLS to take it to the nucleus, and then the NOS terminator. For the gRNA, it's the guide sequence, like the 20 nucleotide, and then the gRNA scaffold, and then the poly-T sequence, which gave a termination signal for the U6 promoter. So the difference between here, this promoter and this promoter, this is a termination signal which will keep it inside the nucleus. But here, the termination signal will take it from out of the nucleus, and it will go to the cytosol, and the protein will be made. And then, so different RNA binding protein will bind to this transcript, and different single-stranded RNA binding protein will bind to keep it inside the nucleus. And this is the basic structure of the produced material. Like we have the Cas9, we have the gRNA guide sequence, which is complementary to the target sequence. And then we have the scaffold, which is responsible for the activation of the Cas9. So Cas9 have two types of activations. The first one is coming from binding with the scaffold, and the second one is coming from the POM sequence. So when the POM sequence is there, when the scaffold is there, then the Cas9 is activated. But then if this sequence match perfectly, it will make a cut. If not, it will go and it will scan for the right target. And then it was confirmed that how these systems are working in the two different organisms. So they use in Arabidopsis like ATPDS. They use two targets, ATPDS and FLS2. So here you can see that the system show only 5% of the editing, and here just 1%. But when the same system is used in the tobacco system, here you can see that the efficiency is very high in both targets. This means that we should be very careful about designing the vectors for the CRISPR-Cas system. And most of the people initially, most of the people try in Arabidopsis and they failed, and nobody get a real editing efficiency, a high efficiency in the Arabidopsis. And the reason is not only the promoter, but also the terminator they were using in the initial experiment for Arabidopsis. To improve this, different groups then start that why it's not working in Arabidopsis. So then like they select the X cell specific promoter, like to express the Cas9, and a specific terminator, that is the Robisco terminator from the peas, and then these two together, and the Cas9 was ZMS Codon optimized, the plant Codon optimized Cas9, and this system worked efficiently in Arabidopsis, and they use two different U6 promoter. So U626 promoter and U629 promoter to express the guide sequence for two different genes, the T and C and ETC2. So this system confirmed that yes, it can work in Arabidopsis if you change the promoter and terminator. But again, the efficiency was very low. So our group also tried to enhance this system, and we used the meiotic specific promoter. So this promoter is expressed only in the meiosis. And we were thinking that if you can target the genome at the meiosis in the next generation, you can get directly the mutated plants without going to the T2 and T3 generation. So this system worked very well. And here we can see that compared to the original one, which is only 5% mutation, the meiotic promoter, we use 3 different meiotic promoters. The efficiency enhanced very robustly, and here you can see we can reach up to 33% just in the T1 generation. So this system is working very well in Arabidopsis, and also this can be adopted for every plant, specifically if we know the meiotic promoter or the XL specific promoter, and we can express the Cas9 at that stage. Then another system like this also opened the field that you can just target only genes that are expressed in the flowering, that in fluorescence, or shoots, or leaves, or in roots, or even the specific cells. Like in the leaf, you can just target specific cells, and only those cells genome will be targeted. And here you can see that when specific promoter are used, the Cas9 is expressed only in specific cells, not in other cells. The same, like for example, the ubiquitin is used as a control. So here you can see the whole plant is mutated. But when another promoter is used, the genes are only targeted on those tissues, but not as a whole plant. So this means that in the plants, we can target specific tissue or specific cells. Another interesting system that was recently developed was to avoid tissue culturing. Like this is a very good system. And here what they did, like for example, they use two different genes, the BBM and the WOSHL, and they express this from a binary vector, including Cas9. So they just inject infiltrate the agrobacterium expressing the BBM1 and WS, and then the regeneration start directly from the leaves. So no need of tissue culturing of the hormonal balance or these things. You can just cut it and put it in the rooting media, and this system can work efficiently. So this vector system specifically developed to avoid tissue culturing and to do efficient genome editing. Then the next step was the binary vector for the monocots. So in monocots, as I explained, that the ubiquitin is working very properly for the Cas9 and the T-NOS terminator. And you can use the U3 or the U6 promoter to express the gRNAs. But this, normally in the cereals, we need multiplexing, and for multiplexing, a very good system was developed. That is the combination of the tRNA structure with the gRNA. So what they did, actually, they put a tRNA and then a gRNA structure, then again tRNA and gRNA. So what happens that when this transcript is made, the gRNA, multiple gRNAs will be produced because of the two nucleases, that is the RNase-P and RNase-Z. So RNase-Z make a cut here and RNase-P make a cut here, and the gRNA will be released, and the multiplex system can be used very well. And this is one of the most widely used systems in the monocot system, including rice, barley, wheat, and this is working very well for the monocot. So to improve this system, one is the multiplex targeting. The second one is normally we need the repair, not only the editing. So if we can do like the repair, so we design another system that if RNA can be used, like for example, we design a gRNA, and then we put a repair template that can be used for the DNA repair as a template or as a complementary strand, and then this repair can, if we put the repair here, it can be converted into the DNA, and then we put like the cis antisense and cis-sense system, and also we put it in trans, like we put the tRNA gRNA structure, so the gRNA is there and the tRNA is there, and then the repair template is there. So it will be cut here, and the repair template will be available for the repair. This is not the DNA sequence, this is the RNA sequence. So this system worked very well. And then here you can see, if you put the mutation here on the repair template of the RNA, it can move to the DNA sequence, and then you can produce the BS resistance, the drug resistance seeds. This is the wild type seeds, and here you can see these seeds are resistant to the drug. Another system that, because we need the gene targeting and repair, mostly like if a gene is not working or if we can put a specific function to a gene, this is important. For that, another vector system was developed that is like the Cas9 and the Vir2 fusion. So all the system is the same, except we put the Vir2. Vir2 is the agrobacterium protein, which is basically transforming tDNA, which is bound with the tDNA and transfer the tDNA from agrobacterium to the plant. So if you have this Vir2, it can bind with any repair template, and the Cas9 can make a cut, and the repair template will be there, and this can be used for the repair at that site. So like for example, we have a repair template and including the right border for the VIRD2 binding. We have Cas9 and we have the gRNA. And then we use this system to make this complex, and this complex can guide the target with the Cas9, and the repair template will be bound with the VIRD2, and the system can use for HDR, and we can get the homology directed repair. So in the next step, we look for these system that it's working or not in the plants. And here you can see that when this system is, we put specific mutation, which is giving resistance to the herbicide. And here we can see that when this mutation is inserted into the genome of the plant, we can see the mutation is there. And then we can see here that these repairs, like we can enhance the HDR up to 20%, which is a good amount for the plant system. The next system that we just confirm the next progeny of these plants that yes, this mutation or this system, vector system, is working permanently, and we can get the mutation in the next generation. One of the best system till now that is working is the binary vector system. But in plants, multiple viruses can be used as vectors for genome editing specifically. So multiple viruses are available, like they are DNA viruses or RNA viruses, and they can be used as vector for the gRNA expression. So one of the most widely used system is the TRV system. It have two genomes, like RNA1 and RNA2. So what we did, we put the gRNA to be expressed from the RNA2, and then we just put to one leaf. We put this system into one leaf of the plant, and then the virus will move, this vector will move to the whole plant, and the whole plant cells will be mutated, including the germline cells. And when the seeds are there, you can get directly mutated plants without any tissue culturing. So this is a good system for the research purpose. And then you can see that we can get the leaf that is infiltrated as well as the leaf that is not infiltrated but systemic leaf from here, and we can get mutation there, and in the next generation we can see that yes, the mutation is heritable and the system can be used. The only problem with this system was the low efficiency, like it was only 3% of the progeny were mutated. So another group, they really did a great job, and they add actually a mobile signal to the gRNA. The problem with the TRV is that it cannot easily move to the germline cells. So what they did, they put a mobile signal to the gRNA, and now they can, they use our system, but they put just a signal, mobile signal to the gRNA in this vector, and then it can move to the germline cell. They basically put the FT locus signal, which is produced basically in roots and move to the shoots. So it's a 35 nucleotide sequence, which can move from one part to another part in the plants. And then they use this system. And here you can see that actually when you infiltrate this vector system into this leaf, they will move. And here you can see the plants are becoming, they're targeting PDS gene. So the plants, this part is becoming white. And here you can see that the PDS targeting is there. And this is in the next generation. So this FT locus is basically giving the chance to the RNA to move to these cells. And this is a good enhancement to our system. In the next, the same system was developed using a different virus, which is the monocot virus, the barley strep mosaic virus. And the barley strep mosaic virus, it have three-partite genome. It have 3 parts. So what they did, they just put the gRNA, and also they include the tRNA, because the tRNA gRNA structure working very well in the monocots. And also they put the FT signal. So they can get easily, like for example, this is the genome, and they put the gRNA, and this is injected to one part of the plants, and then it's inoculated. It's first replicated in the tobacco, then put to the plants. And as this is RNA, there is no DNA sequence. So they just put the RNA to this one, and then you can get the mutated wheat. And the next generation, and these are non-transgenic because there is no involvement of the DNA. This system is this vector system also works very well. But the most interesting one is the recently published one, where they put all the things, including the Cas9 as well as the tRNA, gRNA structure, into the virus, to the SYNV virus, and they express all the things from the virus. And this is RNA virus. So if we have this virus and this system, this vector system, we can use this one for the non-transgenic genetically modified plants, which is a very big concern. So what they did actually, if you have a plant, you just infect and you clone the Cas9 and gRNA, you just put this virus into the plant, and then you just get because this is RNA virus, it will replicate, and then you can get the new plants from these plants. So this system, or this vector system, is recently published, and I think this have a good future. And the other one that did all these, what I was presenting, this was about the RNA vectors. Then the DNA vectors can be also used because the DNA viruses normally they go to the plant nucleus, and there is a good chance that these can be used to express the Cas9 as well as the gRNAs. So for the monocot, they produce you just need the LIR sequences from these viruses, and then you can put the Cas9 material or the CRISPR-Cas system into this system. And when it's replicate inside the plant cell, you will see a lot of viruses produced compared to the single agrobacterium transformation. Here you have a huge amount of the Cas9 replicons. And the rate of the mutation is very high. Recently, another system, basically the same concept, but another system from the wheat dwarf virus was developed for monocot, where they use the ubiquitin promoter with the Cas9 and the U6 gRNA for the Cas9. But compared to the ubiquitin one, the normal Cas9 one, maybe this system is not tolerated by the virus. And here you can see that without the ubiquitin, the efficiency is high, but with the ubiquitin, the efficiency is low. Ubiquitin is not a viral promoter, and maybe it's not working as efficient as the virus promoters are working in the replicons. And here you can see that the system is working very well for these system. Recently, we are thinking about a virus system, and we are developing this system. So like, for example, you have the CRISPR-Cas system in the plant. You just cut the plant here, you inoculate the plants with this vector here, and then the next shoot will come out because the plants have good regeneration rate, and the progenies will be mutated. So we try this system, and we are working on it. So here you can see that when you inject the Cas9 GP, we can see that the plants become GP, the GP luminescence is here, and here, and here, and this means that the new shoot coming out will be the new seedlings or the new shoot coming out will be expressing Cas9, and hopefully they will be mutated. We just did some sequencing, and we can see that yes, some of these progenies, they are mutated. So if this is working, then it will be really good vectors for the without tissue culturing and no need for the transformation. Again, another problem in the plant system is the transgenics. So nobody likes the transgenic plants. The Cas9 can be translocated from agrobacterium because agrobacterium have the capacity to translocate its protein, virulence protein, to the plants. So it's a natural engineer. And we think that if we can translocate the Cas9, if we express the Cas9 in agrobacterium, putting the VERF signal with the Cas9, then this protein will be produced here, and then due to this signal, it will be translocated out of the agrobacterium. And if it can go out of and specifically if we produce the sgRNA also, so this complex is produced in agrobacterium, and this is as a complex, not any DNA or anything else, like no RNA, no DNA, only RNA and protein, and then it's translocated into the plant cell. And then it will go to the nucleus because it have the nuclear localization signal in the Cas9. and then it will mutate. So if this can work, the plants will be completely non-transgenic because there is no transfer of any DNA. We check this system, it's working very well in Agrobacterium. And then we try it in plant system. And here we can see that up to 9% activity we can see for genome editing. But to enhance this one, we couple this system with the TRV system. So the Cas9 is coming from the Agrobacterium, and the gRNA is coming from the TRV system that we already established and that I present in one of the slides. So the RNA is coming, the gRNA is coming from here, and the protein is coming from here. And then the whole complex is made and the genome is mutated, and here we can go up to 18% of the mutation. And here we can see that the next generation is mutated. Then the CRISPR-Cas-based vector can be used for virus interference because most of the viruses, they are going to the nucleus. And if we direct the Cas9 into the nucleus, we can target the viruses because the viruses are replicating in the nucleus. And if we produce the same complex inside the nucleus, so we can target the virus and we can cut the genome of the virus. So for that one, we just try first the TYLCV and the tobacco leaf curl virus as well as the cotton leaf curl virus. But here you can see that mainly the IR region targeting, the coat protein targeting, and the RCR2 targeting. You just need to express the gRNAs, and then you can see that the virus is mutated in these regions. And there is no accumulation of the virus anymore in these plants compared to the control plants. And you can see that when you are expressing the gRNA against the viruses, you can recover these plants. So the virus vectors can be used to provide resistance against pathogens, specifically viruses, because their genome is directly available inside the nucleus. We also use this system with the Cas13 to provide resistance against RNA viruses, and the Cas13 can process the gRNAs, so you can produce multiple gRNAs against the RNA viruses, and then it can target the virus, and there is no accumulation compared to the wild type, so the virus is not accumulating anymore in these plants. And here you can see the plants are the plants are providing very good resistance against these viruses when we are expressing these system. So in conclusion, the vector selection is the core for efficient plant genome editing. An expression of the CRISPR-Cas system in monocot and dicot have specific requirements. You cannot use the monocot one in dicot or the dicot in monocot. They have specific requirements and they need it. The multiplex targeting is possible. Cas12 by itself, it's processing its gRNA. But for the Cas9 targeting, multiplex targeting, we need the tRNA, gRNA, or the ribozyme. Both DNA and RNA viruses vectors are available, and the deconstructed virus can be used. They are very efficient compared to the normal binary vector system or the translocation system or to the RNP system. Tissue-specific or phase-specific vectors can enhance the recovery of the genome edited in next progeny. And RNA vectors and genome editing reagent translocation can provide non-transgenic editing of the genome. So this is all about my presentation. This is our group. This is our group leader, Magdi Mahfouz. And this is Dr. Harun working with rice, basically. This is Rashid, he is working with the dicots. Khalid also joined recently, and he is also working with rice. The rest of the group, they are the PhD students, and they are really brilliant students, especially Ahmad. He is working now on the diagnostic using the CRISPR-Cas system. Thanks for your attention, and if there is any question.
Okay, thank you very much for this great presentation. Now, do you have any time to continue with us in this presentation, or should we take the questions for you, Zahira?
It's okay. It's okay. I can meet here.
Okay, if you can just stay for Mark's presentation, then we can take Mark's presentation right now, and at the end we can have a general discussion altogether.
Okay, okay. Yep.
Okay, thank you so much. We appreciate your presentation. Now we have our final speaker, Mark Smedley, coming from UK. And now first I need to take his presentation. Give me just 1 second. Ankara yazma. Ekranda paylaşmışsınız.
Nereye paylaştın? Açıldı mı ses?
Açıldı onu.
The pointer da açarsan sevinirim.
Tamam. Orası normal. Burada sadece böyle. Pointer burada değildi ama oraya şöyle. Biraz zor olabilir.
Hazır olacak mı?
Şurada. Oraya bakıp burada.
Tamam. Bak.
Ha yok değil galiba. Tamam oluyor.
Bir saatlikten değiştireceğiz. Tamam mı? Tamam.
You can sit it here. Brief information. Now, basically, now our next speaker is Mark Smedley. He's joining us from our Genome Chapter. He has been transferring plants for a very long time. We are really good at wheat and barley transformation. So today he will talk about us. He will give us a brief example, like a case study on RNA design and cloning.
Thank you. Can everybody hear me? So, I'm going to give a on edit in with.
Yes. Okay. I think now it'll work. Okay. That's it.
Okay. Take. So this complex weak genome came about by
two wild species, two wild grass species, crossing about 400 years ago. This gave us the wild Emina wheat. And then about 10,000 years ago, it crossed with Cowshii to give us our bread wheat, or was domesticated and became our bread wheat. And likewise, the wild Emina was domesticated and became our pasta wheat. So this all actually happened southeast of Ankara in Turkey. And this sort of played out like this. So the wild Emina cross was as I said, southeast in Turkey. There was two domestications, one here in Turkey, and one the southern domestication. And then moving over further to what is now Iran, the crossing with Cowshii came about, and this was then domesticated in this area. So this complex genome means that we've actually got 3 copies of every gene, and there's 97% similarity, which can cause problems, but can also make targeting all 3 easy sometimes when designing guides. Similar expression pattern mostly across the 3 subgenomes. There's huge plasticity, and it's a dynamic. And there's a buffering effect, which for genome editing, if you're doing knockout, then invariably you want to knock out all 3 genomes. So we actually see with some genes that we actually get an additive effect. So if we knock out A, we will get a knockdown. If we knock out B, we will get maybe a third less expression. But we need to knock out all 3 genomes to completely knock out a gene. So we've already designed this wheat. We're using 2 whole 3 promoters. We're using the wheat, the TA use promoter or TA promoter, and our guide RNA is cloned downstream of that. You can see the scaffolds in here in the blue. Is where the Cas actually locks into the guide RNA. And for transcription start, we generally start with the U6. We start with a G in our guide, and with a U3, we generally start with an A. And if in the guide sequence there isn't actually a G, we can add an additional G. So designing guides, guide RNAs in wheat. So if you want to target a single copy of a gene, and we have done that in the past. I think our first publication in wheat was targeting just 1 single copy. And I think Shadia will talk about that tomorrow. The best way, or the easiest way that I find to do it, is to actually do an alignment of all 3 genes, looking at the coding sequence, and I tend to design two guide RNAs I tend to design 4 guide RNAs and clone 2 guide RNAs in per construct, and 2 constructs per target. So when designing a guide RNA for to knock out a single copy of a gene in wheat, I will line the sequence, and then I will look for differences. I will look for differences in the sequence, and I will then design the guides in around those differences. However, if we're planning on knocking out all 3 subgenomes, then I tend to align the sequence, and I then look for similarities. I look for areas of homology. I can then design my guides, and these guides will target the guides to the right of the screen can actually target all three copies. So I would then choose this is a CRISPR wheat. This is an online tool. The nice thing about this is that it's actually linked to the wheat genome, which is the Chinese spring genome, and which means that it look for off targets. It can give an off target score as well as an on target score. And there is a tick box when you put in your sequence. There's a tick box which basically says, do you want to target just that one gene, that one subgenome, or do you want to target all 3 copies of the box? Will tell you the guides that target all 3 copies of the gene. Now, I would like to say that generally don't work with Chinese spring. So invariably, what we used to do was actually PCR through the gene of interest in our target species, which was Fielder, which was the transformable, then design our guides on that sequence, or check our guides on that sequence to make sure that it's okay. Charlie's going to talk tomorrow about a number of different wheat species that we're transforming and genome editing in, and we now have the sequence, the genome sequence of Fielder, Cadenza, Kronos, Paragon, and several other wheat species as well. So you know, it's getting easier. All tools are. So now we've sort of designed our guides. We then need to think about cloning strategies. And we're using a Golgate system for ours, and I'll just give an overview, and then I'll show you some of the wheat vectors specifically designed that we're using. So Golgate cloning. Yeah. Just go back. Sorry. I'll just have to talk through. Okay. So Golgate cloning relies on these type 2S restriction enzymes. Now the beauty of these are that they have a recognition sequence, and then they miss a base, and then they cut, leaving a full nucleotide overhang. Unlike sort of traditional cloning, you can use 1 or 2 of these type 2S restriction enzymes, but you can have a multitude of overhangs, and you can choose those overhangs as you go. So Golgate cloning basically is a one-pot cloning method, and you would have your promoter, your coding region, your terminator. You would put this together with your type 2S restriction enzyme. In this case, we're using BSA1, ligase, and a acceptor clone, acceptor plasmid, such as this Golgate vector here, which has the LAC-Z for blue and white screening. The tube then goes into basically a PCR machine. It goes through rounds of 37 and 16, so cutting the DNA, ligating the DNA. Because the components are actually cut out of these first plasmids, leaving the with these overhangs, and leaving the enzyme recognition sequence within the vector backbone. Once these have ligated and they're stuck, they are stuck kind of permanently. The enzyme won't come again. So if you then put these another restriction enzyme together, in this case I've put BPI, this can then take out the cassette that you've just built and clone into another acceptor vector. By using this method, you can actually make very complex vectors very, very quickly and very easy. It's a very robust system. So we're actually using what we call the MOL-close system, modular cloning system. It has set overhangs, and it's hierarchical in its system. So the level ones would be your individual promoters, coding regions, terminators. Your level twos are your transcription cassettes, sort of put together already in these into these level ones. These each have a position. So the overhang, when they're cut out with BPI, they will their overhangs will align them into a level 2 acceptor, and this will be your final finished vector. And you need a linker here which links the very last component to the acceptor, to the right border of the acceptor. As I said, we're using the MOL-close system, and this has a number of set overhangs, which means that you can easily put together your promoters, your coding regions, terminators in one pot reaction. So with wheat and cloning your guide RNAs, we're doing a very similar system to the speaker, in that we order 2 complementary nucleotides guide RNA sequence, and we have our overhangs are in red here. We then do a Golgate reaction, and these are then dropped into level 1 position. And I will explain a little bit in removing and this very efficient percent white colonies. Once your level 1's made, you've got your guiders, and then you need to put together your level 2 vector or your binary vector, which the positions on these Golgate vectors is basically position from the left border going across the right border, which is position 7. And as you can see, for wheat, we will have our selection. We always put the selection gene close to the left border. That's basically because the tDNA goes in via the right border, and you can get truncation at the left. So obviously, if your selection goes there and you get truncated selection, you won't select those plants coming through. We're actually using the rice actin promoter driving the hygromycin selection for wheat, but we have started using the switchgrass UB2 driving the high selection as well. To drive the Cas9, we're actually using the rice ubiquitin promoter. We find that a little bit stronger than the maize ubiquitin promoter, and we're getting better results. And in position 3, position 4, this is where we've previously cloned our guide. And of course, you have a linker here. These all go together in a one-pot reaction vector. This is our level 2 PGG or P golden green gate. This is a vector that I made. It's P green based, but I've put an additional left board in it, and I've golden gate compatible. And all the vectors are actually available on AdGene at the John Innes Center Crop Transformation and Genome Editing page. Your final vector will look like this, and it's just then a case of sequencing, putting into agrobacterium, and then transforming into wheat. So, as I said, Dr. Shadi Hayter is going to be talking about the wheat transformation tomorrow, and we'll show some of the genome editing results that we've actually got. The vectors and the transformation protocol are both together in current protocols, and it's open access as well for those who want to go into more detail. So once you've transformed your plants, you're getting little plantlet back. You then need to start to think about screening. Hopefully, you might have actually thought about screening before that, but it's not always the case. With wheat, because of the complex genome, there's several ways that you can do it. The easiest way that we find is to actually design genome-specific primers that will amplify through each copy of the gene, and we just directly sang a sequence for across the target area. We also run a little bit out on a gel because we're using a 2-guide system. If the guides have worked simultaneously and you've got a large deletion, you'll usually see that on a gel, and it's a nice way of actually following edits in the next generation, in the T1 generation. So once you've got your sequence back, you can look at your sequence trace, and this is a typical kind of edit that we would see. So here's your guide RNA, here's your PAM. You expect Cas9 to cut between the 3rd and the 4th base from the PAM motif. So you'd expect to cut here, and it's just a case of looking through the traces and looking where the guide is, and you see this this drop in signal, and again, these kind of double peaks. And this is basically where two products are then going through the sequencer, and the sequencer doesn't know which bases to call. At the moment, we are using next generation sequencing to look for edits. And we're finding this very, very accurate. But with Illumina sequencing, the products are very small, about 150 bases. And if you get a large deletion, it can be problematic trying to identify those with next generation sequencing. We're now looking at nanopore and setting up in-house nanopore sequencing, which will give us the best of two worlds, if you like, in the sense that nanopore will be able to sequence a 500 base pair product, and you will have the accuracy of next generation sequencing. So, so I've basically talked through here with knockouts. This is kind of the basic knockout system that we're using. It's the most popular, but we're also doing so there's and I'd just like to finish up now just by thanking the people or recognizing the people in our group, particularly Shadia for the wheat transformation, which is certainly the best that it's ever been at John Minns. I'd also like to thank Martha Clark, Shadia's RA, who helps me quite substantially with the cloning, and also Maria and Macarena for all their plant work. I'd also like to recognise some of our collaborators in Christobal's group, in Graham Moore's group, and Jorge and Juan in UC Davis. And thank you very much.
Thank you very much for the presentation. Now we can take the questions for all three. Any questions from the physical participants?
Hi.
Thank you for the presentation. So actually, I can ask you also like you run also. So did you try any root specific for the expression of time? Because people now using yes, promote maybe, but like any good promoter for root specific or any other form specific promoter. Did you try that one?
No, we haven't. I mean, so if would you just want to produce like a chimeric plant, so producing your in the roots, maybe knocking out a gene in the roots, but not knocking it out in the top part of the plant? Or
my aim is to overexpress some genes in just food. So
right.
in CRISPR system, so just for example, I will overexpress some gene in just root. So I'm going to use root specific promoter in case time.
Okay.
In literature, so I don't see any strong root specific promoter. So
I don't actually know of any strong root specific promoters. It would probably be somebody who's working on sort of bacteria and fungi with the roots would probably know much more than me on root specific. But you can always enhance promoters or enhance your Cas9 with intron enhancement. I mean, it's been shown in Arabidopsis that they put 13 different introns in the Cas9, and expression was much higher. I do know that a lot of weaker promoters in wheat and in barley, we've actually used the leader intron from the ubiquitin promoter, the maize ubiquitin promoter. It's 2 kb long, but 1 kb of that is actually a leader intron, and by positioning that downstream of a weak promoter, that actually enhances the expression of a weak promoter.
Seed cotton, actually knows that wanted to reduce the cost of phosphorus in the cotton seed so that it can be utilized by so what we have done was that so seed stuff. So the expression of Cas9 was is required for the cotton on the other part to give protection against it. So we have done this. What is efficiency? So efficiency was because it was expressing at the same the level of expression of phosphorus in the leaves, and the upper part was the same part in the seeds.
In order to elaborate this question, can we ask exactly the same question to Zahir Ali as well? Do you know any promoters that are specific to the roots that can be used?
I don't know exactly, because we are mainly working with the progenies, so I'm not working with the development. But definitely, for example, Ikram, she is one of the leading scientists in developmental biology. So I think you can look at her work, and definitely she did some work with the root promoters. Specifically with the auxins promoter, I think the auxins promoter, producing promoters, they are basically root specific promoters.
Okay, now we can take one more question.
So I have a question for Zahir. Please available. Zahir?
Yep.
Can you hear me? So my question is that, what do you suggest, the system for vegetative crop like potato and cassava and banana? So, what could be the ideal system for every genome level? Because, there's no seed system, so obviously we need to remove the pests. So what is the solution?
Can you repeat the question? I didn't got the exact.
This is about vegetatively grown crop like potato, cassava, banana.
Can somebody repeat near to the mic? I cannot hear the voice.
Sorry, I have to translate. Okay, Zahir, my question is about genome editing in vegetatively grown crops, so like potato or cassava or banana. So what system you suggest? Because in case of crop where we have a system available.
Yep. Actually, I present some of the. I will just look at that back. I think the translocation system is working better for the especially vegetative one, because we cannot remove the transgene from there. So the agrobacterium mediated translocation system is working much better for the vegetative system. So I think if this can be applied like in potato or vegetative crops, I think it can be used very well.
And
one more question to Mark is about cis-acting elements to actually not to knock out, but change the expression levels.
That's also possible.
Yeah, sure.
So maybe I can answer both questions actually. So the what we're actually doing with wheat in one particular case is that we've got a heat shock promoter driving a Cre recombinase, which has an intron in it, and we just put lots P sites either side of the tDNA, and then heat shock the material when we want to remove the tDNA, and the tDNA is recombined out. It does leave about 16 bases, I think. So it's not absolutely clean, but that's almost as good as clean. I mean, 16 bases is less than an oligo. Sorry, could you repeat your question again? Yes, we are at the moment actually. So we're actually targeting promoters, and we've we're multiplexing, and we've designed 4 and 6 guide RNAs for several different promoters, and we're hoping that the guides won't be very efficient, so we'll get a lot of variety in those promoters coming through. So obviously, if the guides were very efficient, and we're targeting maybe 2 kb of the promoter region, if they were very efficient, then they would just take out 2 kb of the promoter, and and that would be it basically. But what we want is to target. We're targeting different kind of sites within that promoter, hopefully to change the expression levels either up, down, or in time, or spatially or spatially.
Okay, any other questions from the audience? One more general question.
This one works better.
Yes. I don't know whom to turn my back. So more general question. If you number the difficulties working with wheat, regarding CRISPRing wheat, what would be the most difficult, the biggest challenges working with wheat while doing the CRISPR compared to Arabidopsis or rice, for example?
So I think actually targeting just a single gene within one genome, that can often be quite problematic. Certainly with 97% homology, actually choosing guides is very, very difficult when you're doing that. But again, the tools have increased, so we're trying different Cases and different Cas proteins within wheat. They have different PAMs. So if one PAM is sort of available in one particular genome but not in another subgenome, then that will obviously make it a lot easier. So that's quite a difficult thing. The other thing is that with crops, they tend to be genotype dependent. So you can only transform perhaps one type of one variety, if you like. So with wheat, it was always Bob White that people would transform, and then Fielder came along and people started to transform Field because it was more efficient. And Kronos transformation is fairly inefficient, but that's a tetraploid wheat, a pasta wheat. Well, there's a lot of resources in there. Now we have a technology, a GRF technology, Charlie's going to talk about tomorrow, that has actually broken down all those barriers. So transformation efficiency for tetraploid wheat now is about 80%, and the same for many of the other wheat varieties. We're getting high transformation efficiency. And obviously, if you can transform different varieties and produce a lot of transgenic plants, then it's easy to try and, you're just sport for choice for actually looking for those edits, and if you're looking for knockouts, you then just look for the large deletions. You don't bother with the small ones. So yeah.
Okay, are there any questions? Are there any questions from the physical audience? Can you come to the stage to ask that?
Hi, Mark. I have a question. When you are using a modular cloning system, how do you choose the promoters and terminators?
How do I
choose promoters and terminators when you are using modular cloning system?
Mark, can you repeat the question?
Yes. How do I choose when I'm using a modular cloning system? How do I choose promoters and terminators? It really depends on the species that you work in. So if I was working in, I would use promoters that are dicot promoters that are specific for those. We actually find that in cassava mosaic virus, terminator across the cereals, across the 35S terminator or the NOS terminator, and they work quite well in all the different species. But it's not so much choosing the promoters or the terminators because of the modular system. It's choosing them because of the plant variety that you're actually working in. So in brassica, for the pole 3, we're actually using the U3, the U6 26 to drive the guides. We find that works very well. So it's just really down to the species that you're working in. Most of the things that work in wheat will work in barley, but in barley selection, a very strong cereal promoter driving the selection, the hygromycin. We'll just have the 35S driving the hygromycin, but that is intron enhanced with the Cat1 intron.
Now, I think we can take a few questions from the online. Okay, actually, this chat is also recorded. I think we can share that with you. Now, let me just 1 second. Basically, Dr. Hansel mentioned about 2 papers about some root experiments in rice. So when we share those box things, so you can just see those comments. Now let me see if there are any other questions. Yeah, I think there are no other questions from the online audience. Now I have one other thing related to the root specific promoters. I think there was a paper published from Ben Fee's group long time ago, I think in 2008. At that time, they generated some RFP constructs, and those constructs actually are defining the promoters were from different parts of the root tissue, and they were just comparing their activities. So maybe if you can find that paper, that can help you. Okay, we have one question. Are there any chances of allergy development during wheat transformation? Any comments?
I think that should be addressed to actually. I do know somebody who worked with barley for quite a long time and ended up getting allergic to barley. And imagine that if you do work with one particular, perhaps likely to get censored. That kind of answers the question.
So we came to the end of the today's lectures. I really thank all the participants, all the lecturers. Thank you very much for your efforts. Tomorrow again we will start on at 9 o'clock Istanbul time, and then we will continue the rest of the lectures. Thank you very much.
Thank you.